All right, we're going to finish out our notes by talking about active transport. So the diagram right here shows you the passive transport that we've talked about, diffusion, facilitated diffusion. Now this is passive transport of solutes. If we added passive transport of solvents in here, we would also see osmosis. And now we get to see active transport. So a couple of things that you might recognize that are same but different. So we still have to use a protein. So here is a protein, just like we saw with facilitated diffusion. Notice the gradient. So instead of in passive transport, we were moving from high to low. In active transport, we're going to reverse it, and we're going to move from an area that is already high concentration. We're going to try to put more into it. So we're going to go from low concentration into a high concentration environment, which is challenging. And so we need right here, we need a little bit of energy, ATP. ATP always binds to the intracellular domain of the transport protein. So it always attaches to the protein on the inside of the cell. So there's two broad categories of active transport. The first refer to pumps, which are really transmembrane proteins, and that's what we see right here in the diagram. These pumps require ATP, right? That is cellular energy, to move a solute up its gradient or against its gradient. I've seen both kind of terminologies, and so I want you to be able to recognize both as active transport. So up the gradient would be from an area of low concentration to high concentration, moving up. Against the gradient is sort of that idea of fighting against the natural passive diffusion of taking something that's really concentrated and spreading it out. Instead, we're going against that natural law, and we are taking an area that's already concentrated and trying to put even more into it. So both of those, up or against the gradient, indicate from low to high concentrations. A specific example that we're going to talk about is the sodium-potassium pump. That's sort of a classic example of a membrane pump that is actively transporting sodium and potassium against their gradients in a particular cell type. So let's look at the sodium-potassium pump. So sodium-potassium pumps are in many places, but they are very, very, very common in this cell. So this is a neuron. We've seen a neuron before. These are the primary cell types in your nervous system. And these pumps are embedded on these little tiny extensions up here called the dendrites. So the whole thing is a cell. It's wrapped in a membrane. So even though it looks funny, it does still have a membrane on it. And these pumps are clustered embedded in the membranes of these little short projections up here. Here is a little GIF of the sodium-potassium pump. So you can see a couple of things. You can see these little pink dots are the sodium, and then the little triangles are the potassium. And so you'll notice that as the pump operates, 3 sodium are pumped out and 2 potassium are pumped in. This right here is a phosphate off of the ATP, the adenosine triphosphate energy molecule that is facilitating the movement back and forth of that pump. All right, so let's talk about or write down some information about the sodium-potassium pump. There it is, found in the membrane of neurons. And after a neuron generates an electrical signal and passes that signal down its body, everything's sort of out of whack. And so particularly the sodium and the potassiums, which help the neuron generate the electrical signal, need to be put back into place. And so this particular pump is really sort of an effort to reestablish quote normal so that the neuron can fire again. So this is something that kind of comes in at the tail end of the neuron generating an electrical signal, sending it down its body. Now it needs to reset so that it could be ready to go to receive the next signal and send the next signal. When the sodium-potassium pump is working, so here's back to the diagram, active transport, 3 sodiums out. We'll look at all the sodiums that are already out here. So putting more into an already crowded environment, 2 potassiums in when there's already potassium in here. So putting more potassium back into an already crowded environment takes ATP. So it's going to move 2 K plus, that's potassium, into the cell. That's against the potassium's gradient. And it's going to move 3 sodium out of the cell, which is against the sodium's gradient. And because of that, we need energy. The second big category of active transport actually doesn't have to do with a gradient necessarily. So this is active transport requiring energy, not because of shifting molecules up or against their gradient, but because we are moving in large quantities or large particles into and out of the cell. So this is called bulk transport. Bulk transport, when I think of bulk, I think of stores like Costco and Sam's Club that you go buy large things or large amounts of things. That's what we're going to see. We're transporting large molecules or large quantities of molecules into and out of the cell. So I know that there's not a ton of room on your notes. You can sort of write it off to the side before we jump into our first type of bulk transport, endocytosis, but you do need a little bit of an understanding that bulk transport is about moving large molecules or large quantities. And as you can see in the picture, all bulk transport requires the use of a vesicle to move the substances into or out of the cell. So that sort of needs to be written kind of somewhere around the word bulk, just to help you remember what bulk transport really is. And then the first word that you're going to see is exocytosis. And so there's 2 broad categories of active transport, moving things out of the cell and then moving things into the cell. So if you watch on the screen, this is a diagram of exocytosis. So we have some particles, solutes that are within a vesicle, and that vesicle melds with the membrane and pushes those substances out of the cell. So this is transporting solutes out of the cell, and it can even be larger particles. Solutes is sort of a generic term here. Doesn't have to be the smallest of dissolved solutes. It can be larger, but the key here, exo, we're going to move them out of the cell. And that happens by merging the vesicle with the cell membrane and releasing the contents of the vesicle into the extracellular environment. The opposite we see in this diagram is bringing something into the cell. And so that is endocytosis. And so there's a couple of things transporting substances into a cell. It's really the reverse of exocytosis. So the cell membrane, as the molecule or the thing that's going to come into the cell approaches the membrane sort of pinches off a little bit of itself to create a vesicle and allows that substance to move in. And there's 2 broad categories. When it comes to endocytosis, we break it down into, are we bringing in sort of large, quote, food molecules, or are we bringing in dissolved, quote, drinking molecules? So yes, it's like a reverse exocytosis. Here are the types. So there's 3 primary types. Let's talk the first 2, which are the most common. Pinocytosis is when we're bringing in dissolved solutes. So pino is really cell drinking. The pino refers to almost like you're taking a drink. And so if the cell is drinking in or bringing in dissolved solutes, that specific type of endocytosis is given a more direct name called pinocytosis. What we saw in the previous slide, the little moving GIF, that is a cell eating or taking in more of a solid substance, which is called phagocytosis. And so here in the diagram, you see just those two, pinophagocytosis. Up here, you see bringing in a whole phagocytosis, bringing in dissolved solutes, pinocytosis, but you also see the third one. And so the third one is called receptor-mediated endocytosis, and there's a whole section of notes specific to that. So we'll see that here in the next slide, the actual content for this section. So receptor-mediated endocytosis is, as it sounds, a molecule must bind with a particular receptor on the surface of the cell, and that triggers endocytosis. So if we jump back up here to this diagram, these little green molecules, these are protein receptors. So remember the little surface carbohydrates and the surface lipids that are the glycoproteins, the glycolipids that can act as receptors on the surface of a cell membrane. Here you're looking for the correct molecule, which in this model is the star, binds to the receptor. And once that binds, the process of endocytosis is triggered, the membrane creates a vesicle, and in comes the whatever particular molecule that is. There's a really good example of this process of receptor-mediated endocytosis with a condition called familial hypercholesteremia. And so over here's a tiny little infographic to get us started. Familial hypercholesteremia is also called FH. It is a genetic condition, and really it's a condition that's connected to high cholesterol, high blood cholesterol. But it is, as the infographic indicates, it really is genetic. So there are some people that have high blood cholesterol because they have really poor diets and unhealthy lifestyle habits. There are also other people who, from the day they are born, are genetically inclined to have high blood cholesterol no matter what they really do. And so that's what I'm talking about here is the genetic high cholesterol. So we're going to write a little bit about FH. Before we do, I want you to see the video. Now, I've chopped a portion of this video. We're not going to watch the whole thing. So in addition to us seeing that it is a genetic condition, I want you to really pay attention to, they're going to briefly review cholesterol, which we know is a lipid. And they're going to talk about the two different kinds of cholesterol. And we have very generically called it good and bad cholesterol, LDL and HDL cholesterol. But pay attention to what they do, and you will find very quickly the connection. So this is why we're watching it. We're watching to see the connection to receptor-mediated endocytosis. So let's watch the video first, and then we'll take some notes here.
With familial hypercholesterolemia, familial means the disease runs in families, so it has a genetic predisposition. Hyper means excess, and lastly, cholesterolemia refers to the level of cholesterol in the blood. So familial hypercholesterolemia is a genetic disorder associated with high levels of cholesterol in the blood. Now, cholesterol is a lipid molecule, so a type of fat, that normally helps maintain the structure of cell membranes and is a precursor to steroid hormones, bile acids, and vitamin D. There are two main types of cholesterol: LDL, or low-density lipoprotein, which is sometimes called bad cholesterol, and HDL, or high-density lipoprotein, which is sometimes called good cholesterol. But good and bad is overly simplistic, and like with all things, the subtleties matter. LDL is produced by the liver, and it carries cholesterol out to the rest of the body. If all of the cholesterol from LDL is not completely distributed to the peripheral cells, then HDL brings some of that cholesterol back from the peripheral tissues and sends it to the liver. Now, what makes LDL bad and HDL good is that whenever there's a high blood concentration of LDL, the LDL can be ingested by macrophages that sit along vessel walls, forming atherosclerotic plaques. Over decades, large atherosclerotic plaques can lead to myocardial infarctions, strokes, and peripheral vascular disease. That's why we want to keep LDL blood levels under control. On the other hand, HDL can remove cholesterol from cells, and that can help reverse the process of atherosclerosis. Now, our body usually keeps LDL cholesterol levels in check by clearing our excess LDL from the plasma. This is mainly done by the LDL receptors present on the surface of the liver cells. First, the LDL molecules bind to the LDL receptors, which are clustered in specialized regions of the cell membrane called coated pits. After binding, the coated pits, along with the receptor-bound LDL, are internalized by invagination, and they form coated vesicles inside the cell. Next, the LDL receptor releases the LDL in the cytoplasm, and it gets recycled back to the cell surface. At the same time, the coated vesicles fuse with an intracellular organelle called a lysosome. Inside the lysosomes, LDL molecules are enzymatically degraded and free cholesterol molecules are released, which then cross the lysosomal membrane to enter the cytoplasm. From the cytoplasm, free cholesterol can be used for cell membrane synthesis and other metabolic processes. With familial hypercholesterol,
I really messed up. Okay, I wanted to pause right here for just a second and make sure before he moves forward that we are good with, oh my goodness, so sorry, that we're good with this diagram right here. So here's receptor-mediated endocytosis. This is our bloodstream, right? Up here, this is where our cholesterol will circulate. And then this tissue, which I thought, let's go back up. Yeah, is the liver. And so we want to pull cholesterol out of our bloodstream so that it does, particularly the LDL cholesterol, so that it doesn't form those plaques that we saw just a couple of seconds ago. The plaques can lead to a heart attack, to strokes, and something called peripheral vascular disease. Bad things. So we want to pull the LDL out of our blood. Well, this is how you do that. The liver, so this is going on in the liver. The liver has these receptors on the surface in certain little pockets. And when LDL binds, it pulls that LDL into the body. It will then release the LDLs into a little vesicle, and a lysosome will come and degrade those LDLs and then use those little bits and pieces to resynthesize components of the cell membrane to build in more cholesterol in the next membrane of the next organelle that's being formed in the body. And then this ultimately goes right back out to the surface, and we return those receptors back out to the surface. So ultimately, the more that we do this, the more that we are bringing in just LDLs, because that's all that binds to these specialized receptors in the liver cell. And the more LDLs that we bring into the liver, the less are circulating out in the body. So we have to make sure that we understand this diagram, because he's getting ready to talk about the different categories of familial hypercholesteremia. So this is how it works normally. And here comes several of the genetic implications to altering this normal pathway. Oh my goodness.
plasm. From the cytoplasm, free cholesterol can be used for cell membrane synthesis and other metabolic processes. Now, in familial hypercholesterolemia, mutations occur in the LDL receptor gene. There are over 900 different types of mutations that can cause familial hypercholesterolemia, but luckily they've been grouped into 5 major classes. Class 1 mutations mainly affect the quantity of synthesized LDL receptors, so there are less available receptors to take up LDL from plasma. Class 2 mutations affect the intracellular transport of LDL receptors back to the cell surface. Class 3 mutations affect the binding of LDL to LDL receptors. With Class 4 mutations, LDL receptor proteins can bind LDL efficiently, but they're not located in the coated pits. So even if they bind to LDL, the internalization of LDL fails. Lastly, Class 5 mutations affect the recycling of LDL receptors to the cell surface, which ultimately lowers the total number of functional LDL receptors present on the cell surface. Now, all these mutations have one thing in common. That is, they decrease the clearance of LDL from the plasma, which results in accumulation of LDL cholesterol in the circulation. These excess cholesterols are then engulfed by the macrophages and vascular walls, resulting in premature atherosclerosis.
Okay, so now let's talk about what are we going to write down in this last section of our notes about FH. So first, it's a genetic condition, and you saw in the video there are a lot of different possible mutations that can target different spots in the normal pathway of how cholesterol is taken in by the liver. Here's what typical looks like. So typical LDL levels, right? LDL, that's our bad cholesterol. Typical LDL levels are decreased by receptor mediated endocytosis, removing it from the blood and bringing it into liver tissue. That is normal. So in FH, you have to understand normal to understand how the mutations negatively impact. So in FH, in one of those different classes, and we don't have to know all the different classes of the condition, but we do have to know the net effect is reduced intake of LDLs. And so now that you are not taking in as much LDLs, it is still out in circulation in your bloodstream, which causes these plaques to build up in your arteries, which then can lead to the conditions that they cited in the video, primarily the ones you would be the most familiar with: heart attack and stroke. So again, really easy connection to maybe see receptor mediated endocytosis. When we need to pull something in this case LDLs, we need to pull it into the liver, and we make sure that it is pulled into the liver by allowing it to bind to receptors on the surface of the liver. And when it binds, it triggers the endocytosis, removing it from the bloodstream. All right, that's your last note. So when you're done, you are good.